And so when I begin my lectures, I like to begin at the beginning, the very, very beginning, the birth of the planet, and point out that the first and most successful organisms on the planet are microbes. They're bacteria. They have been around the longest. They are the most successful. They're also the most numerous, the most diverse, and ubiquitous. Everywhere we look on this planet, we can find microbes. They have evolved and adapted to live in the most extreme of environments. That can be everything from an acid mine to the incredible pressures at the bottom of the ocean, to incredible extremes of heat and chemical exposure, for example. So they're adept. They can live in the most extreme environments. And as I said, they are everywhere. So we, as well as every other biological entity on this planet, have evolved in a microbial soup. And in fact, we haven't just evolved in microbial soup, we've co-evolved with microbes. They live in and on us, and we actually rely on them for functions that we ourselves do not encode in the human genome. But how have we studied microbiology? Well, we've taken a very reductionist view traditionally. What we've traditionally done is taken microbes out of their environment, grown them under feast conditions in laboratory media by themselves, and studied what they do. And nothing could be further from the truth of how these microbes exist. They're actually quite social. They live like us in diverse communities. They communicate with one another. They use small molecules to sense who's in their neighborhood and respond to those microbes that are in their neighborhood. So how do we get at these organisms, many of which we've never cultured? We don't know how to grow them. We don't know what they eat. We don't know what they subsist on. We turn to molecular tools. So the kind of workhorse of the field of microbiome research is using DNA-based methods to identify microbes without ever having to grow them or isolate them from a sample. We'll take a sample, we'll extract the DNA from it, and remember that DNA comes from every microbial cell in that sample. They could be various different types of microbes. And then one approach we have is really targeting specific genes, like this one, called the 16S ribosomal RNA gene. This is a gene that's only found in bacteria. It's not found in any higher organisms. And it's a great biomarker gene for identifying which bacterium it came from because it has these regions in the gene that are really highly conserved across all known bacteria. So we use those highly conserved regions to kind of anchor an assay that we have to basically make copies of the region in between. And the regions in between those really conserved regions of the gene are what we call hypervariable. There the sequence varies, and it varies depending on which bacterium the gene came from. So we can make lots of copies of these genes and then sequence the hypervariable region to figure out who it came from. And in that way, we can generate like what you would think of as a fingerprint. Which bacteria are there, and in how much of each bacterium is there? What's the relative abundance? And this is really useful for comparing across very large cohorts of samples where we just want to know who's there and how it differs, for example, in health and disease. We've got a similar tool for looking at fungi. There's a lot of various regions we can look at with the same type of technique, but we tend to use this one called the interspacer region 2. Again, we amplify that piece of the genome from fungi, sequence it, and then we can tell which fungus it actually arose from. And in that way, look at fungal communities and how they're composed, who's in that fungal community. But the tools for assessment of the microbiome have rapidly evolved and expanded in capacity over the last several years. And we now can, instead of just looking at a biomarker gene, actually take all of the DNA we extracted and sequence all of that DNA. And then put those pieces back together, basically reassembling the genomes of all of the microbes in that sample. And this is no small feat, as you can imagine. I joke, although it's not really a joke, and I say it's like me handing you War and Peace by Tolstoy in pieces and asking you to put it all back together in a legible form. That's the computational capacity that we need is immense to do this job, but something that we have developed very rapidly over the last several years. So while biomarker gene sequencing tells us who's there, shotgun metagenomics tells us the genes that these organisms encode and what they have the capacity to do. But we've pushed this field even further. We can also extract RNA from a sample. It's another type of nucleic acid. And it's the transcription of those genes. So it's what is that community of microbes actually transcribing off their genomes? How are they responding to the current conditions? And we can sequence those pools of extracted RNA by sequencing also, and we call this metatranscriptomics. It gives us a snapshot into the genes that are being expressed at the time of sample collection by organisms in the microbiome. But what's even more exciting is that we can look even deeper. These are all next-generation sequencing-based tools to look at microbiomes. We can also use mass spectrometry, the capacity to identify small molecules. And we can use this to look at protein pools produced by the microbiome, to understand the proteins that they produce, and that includes, remember, all of the enzymes and catalytic functions of the microbiome. But what's nearest and dearest to my heart is metabolomics, looking at the small molecules. Remember I said that that's how microbes communicate with each other. In fact, that's how cells communicate with each other, irrespective of their microbial or host. And this, for me, is the lexicon that governs microbial host-human host interactions. And this is where we think the next frontier, and we're already realizing the next frontier in this field really lies. So the application of these tools, and in particular DNA-based tools, has massively expanded our view of bacterial life on this planet. This is the tree of life. This is everything. We're down in one of the little branches down there on the bottom with the eukaryotes. These up here on the top are all bacteria. And this is a study that was published in 2016. Everything in purple are brand new bacteria that were identified in this study alone with molecular methods. So we have immensely diversified the bacterial tree of life. And we suspect that this is also true for viruses and for fungi. We just need to catch up in developing the tools for those realms of microbial life. But what these tools have told us is that there is a much broader range of fungi and viruses, particularly those that exist in the human body, than we previously were led to believe based on culture-based approaches. And in total, the application of all of these tools to interrogate the microbiome has left us realizing that we're not alone. We are, in fact, superorganisms. We're a conglomerate of microbial and mammalian cells that have co-evolved over time. And we are colonized inside and out by microbes. This is just simply looking at microbial diversity across the surface of the skin. Anything in red is kind of higher diversity. In blue is regions where there's lower microbial diversity. This is looking with mass spectrometry at the molecules made by those microbes on the skin. And here you can see that even where there's regions where there's not so many microbes present, there's a huge biochemical diversity of molecules that are produced at those sites. Produced by the microbes, produced by the host cells in response to the microbes. There's a rich molecular lexicon occurring at these sites. That's simply the skin, and that's actually considered a very low microbial burden site. We house the greatest burden and diversity of microbes in our gut, particularly in the distal gut. And these microbes are not simply bystanders. These microbes influence how our gastrointestinal cells behave and function and respond to this microbial zoo in the lower gut. And I will say it's not just the lower gut. There are microbes obviously in the mouth and the whole way down through the GI tract. They differ at different sites along the GI tract. And we think that that's because of the prevailing conditions that differ. If you think about in the stomach, the pH is very low. In the lower gastrointestinal tract, there's very little oxygen there. These are strong selective pressures that drive the types of organisms that like to thrive in these distinct niches along the gastrointestinal tract. But I think what's really amazing is to think about how much our microbiome dwarfs our human genome in terms of genetic capacity and genes that it encodes. This is one study of European, Asian, and US populations. Just over 1,200 fecal samples were sampled and examined using shotgun metagenomics. So looking at all of the microbes and all of the genes encoded by the microbiome in those 1,200 or so samples. And what's staggering is almost 10 million microbial genes were found just across those 1,200 or so individual fecal samples. I want to let you think about that for a second. That is incredible. This is an ancillary microbial genome that we carry around with us. These genes are not silent. They are actively expressed. And we rely on these genes for things like digesting our dietary components, for digesting, metabolizing our drugs, in fact, and for informing and influencing our immune response. So these are an important part of our physiology, an important part of what makes us healthy or diseased. And to add to the complexity that this is not just one type of microbiome that we have in one site, we develop our microbiome in early life. We are born with a very simple microbiome that we inherit from our mothers. It either arises in utero or is contributed to through the birthing process. Babies who are born through the vaginal canal have a preponderance of lactobacillus species, which are the dominant organisms in the female vaginal tract. Babies who come out the sunroof by cesarean section end up quite frequently with organisms we find on the skin, Staphylococcus and Streptococcus, suggesting that very early life postnatal exposures influence those communities of microbes that are found in the very early gut. And as we proceed through very early life development, we now know that a whole range of factors influence and shape the types of microbes and activities of the gut microbiome. Things like early life nutrition, antimicrobial exposure, as I mentioned, cesarean section, really strongly influence what type of microbes are there and how they're functioning. We continue to expand the diversity of bacteria that we have in the gut up until about 3 years of age. Around then, the diversity looks like that of an adult, but the functional genes in that microbiome at 3 years of age are quite different from that of a healthy adult. Throughout life, we continue to shape our microbiomes. In fact, I view them as, in adulthood, as a history of your exposures in life. Things like pharmaceuticals, diet, infection, sex hormones, even environmental toxicants can serve as strong selective pressures on which microbes are present and what they're producing, and therefore how they're interacting with the host.